All right, I wanted to go ahead and start early so I could do some announcements that truly are repetitive. So I'm not worried about them being done before 8 a.m. I got a good question today on an email asking if we were still doing lecture online, or excuse me, in person. And the answer to that is always a yes. It's always going to be the rule that we're in person and the exception that I'm online. So if you ever have a question and the student did the right thing, they sent me an email. But if you ever need a quick answer, like if you send it at 5 in the morning and I haven't answered yet, and you were wanting to know if you need to come to camp or if you should come to campus or not, just check your emails and check Google Classroom, because on Google Classroom, I'll always, if there's any updates to anything where, sometimes where I only teach from home, sometimes on a Friday for family reasons or whatever, emergencies. If that happens, you'll have an announcement. So always check your announcements if you need a quick answer for anything, really. Other than that, everything will go as scheduled. Speaking of which, for lab, people are just not doing labs. So starting next week, next week's lab will be online, as I mentioned before. And still, if you have it done, you do not need to attend lab. But now, starting next week, even though it's an online lab, you guys will need to come to lab in person. Bring your computer. The TA will hopefully have computers. But yeah, from now on, we're doing labs in person, even when they're online, because people just aren't doing them. I don't understand why. I told you at the beginning of the semester that you have scheduled this course, or you have a lab either every Monday or every Wednesday from 2 to 3:50 a.m. or p.m. I said, keep that open, because you never know what's going to happen. Just always assume for your schedule that you're going to be in lab for the day you have scheduled for lab. The day you sat down with your registrar or your advisor and said, this is the class I want. So that's the time. So again, next week, if you haven't done the lab and I haven't graded it yet and send it back to you and said you don't need to come to lab, then you need to physically come to lab, even though it's an online lab. Does that make sense to everybody? Are there any questions? All right. And then you can leave the lab. Oh, yes. Question?
So, like, if you have it done, you don't have to go, right?
Right. Yeah. Like you'll know if you don't have to go, because I'll grade it and I'll send or sometimes I won't grade it, but I'll look at it and say, okay, you don't need to come this week.
And everything.
Right. Yeah. Yeah. You'll know if you don't have to come to lab next week. All right. So that's it for lab. If you guys have any questions, send me an email. What else to say about lab? Oh, yeah. So again, lab's over at 3:50. So when we meet for lab next week, you can leave either when you're done or when 3:50 rolls around, whichever comes first. So just because, people are failing, I shouldn't have to force people to do the bare minimum. And like last week's lab, the one with the DNA, that was really easy. You've got a website that says, do this, put in the proteinase, put in this, and this is what it does. And then the question says, what are you putting in next? What does it do? It's literally right there in front of you. And people are still messing it up, like to monumental degrees. The people that did it, that is. There's a lot of people who haven't even done it. So anyway, that's the news with lab. What else? Just overall, man, if you're failing, let me help you. Work, do the work. Quit making basic mistakes. Get some work done. Let me help you if you need to get caught up. Anyway, attendance. The first word for attendance will be marker, like something you would mark something with. And then for those of you online, as usual, just send me an email. Make sure the email is sent to me before 9 a.m. with the words. And you guys in person, if you don't mind sending me an email too, that'd be good. And for those of you three in person, remind me I'm giving you a little bit of extra credit for being in person. Good? All right. Let's jump back into it. To remind you what we're talking about here today is going from DNA to RNA to protein. And of course, you can study this any way you want, but I'll remind you, my suggestion, my advice for the way to study this is sort of like how I suggested it for photosynthesis and respiration. Start with the big picture and then add details. So for example, start by memorizing the fact that DNA provides instructions on how to build a protein. All right, good. You have that memorized. Then remember that it goes from DNA to RNA to protein. Okay, good. You have that memorized. Then remember the words for that. So going from DNA to RNA is transcription. Going from RNA to protein is translation. And then once you have that memorized, then you can start getting into the stuff that we're about to get into, which is the actual process of transcription and translation. Now, remember, what we've already done previously was an overview. So some of the stuff I'm saying today, hopefully you'll remember it from Monday, because it is an overview, but we will be going a little bit deeper. So are there any questions before we get started on this process? All right, and I know this seems a little bit boring. Even as a biologist, I can understand that. But keep in mind, this is the stuff that makes you what you are. Like going from DNA to protein, this is how your body was built. This is you. So you're learning how you were made. Well, that and what we learned in chapter 8, which was mitosis and meiosis. Anyway, transcription. As you should already know from Monday, this part is a review. Transcription is the transfer of genetic information from DNA to RNA. I'm not going to ask you a definition of transcription. Basically, this is what you need to know about transcription, the most basic. It's when we go from DNA to RNA. There will be a test question that basically just says that. What do you call it when we go from DNA to RNA? Transcription. You can ignore that little line. That's just a glitch that I've been dealing with. As you already know from previous discussions, the DNA strands separate where the process starts. Actually, you don't know that yet. I'm sorry, I haven't said this before. But we have said this as far as DNA replication is concerned. Because remember, that's what we learned about before we started getting into transcription and translation. We were learning about how the DNA was replicated in S phase of interphase. And the way it happens is, remember, the DNA opens up because what's keeping those two strands together is hydrogen bonds, which are very weak. The strands open up, the template is there, and then the DNA polymerase comes in, it reads the template, and that's how DNA is replicated. And it's almost the same thing for transcription, right? So the DNA separates. We have another enzyme comes in. It's not DNA polymerase this time. It's a different one, and I'll tell you which one it is later. And it reads the code because, again, the strands open up, and that exposes the bases. Right, those little lines coming off are the bases. So there's an A here maybe, and a C, and a G, right? That's the code. And one of those is going to serve as the template. Which one doesn't matter, whether it's the left one or the right one. But yeah, one of them is going to serve as a template, and the other one will be unused. The RNA nucleotides take their place along the DNA template by forming hydrogen bonds with the DNA nucleotide bases. This is basically, again, if you've already understood how DNA replication works, and this is the same sort of process, right? The cellular machinery reads the code, like down here at the bottom left, it reads this A and says, oh, if there's an A, I need to bring in a U. So it does. And you can't see it because that's horrible. Anyway, and then it sees that there's a G or a C, so it brings in a G, and then that gets hydrogen bonded. And then it sees there's a G, so it brings in a C, and that gets hydrogen bonded. It works the same way as far as we're concerned in this course. Any ideas why I have a picture of a pen on this side? What does that have anything to do with transcription? Why would I be doing that? What kind of reminder am I trying to give you from the conversation we had on Monday? Good guess. I like that guess. Try to remember what I said about writing on Monday. Because obviously a pen has nothing to do with biology. So what did we talk about that had nothing to do with biology? And I was trying to make that connection between the non-biology version of something and the biology version of something. And you could use some context clues as college education.
We talked about translation.
Close, yes. You're getting closer. Because translation is going from one language to another, right? So that was the same conversation. That's all right. I'll remind you guys. Here's a big hint for those of you guys who like to figure out mysteries. We're talking about transcription, so you might guess that it has something to do with transcription because that is what's on the board right now. Anyway, yes, transcription. To remind you, when we first started talking about transcription, I said, hey, what does the word transcribe mean outside of biology? And someone, I think it was Cordone, I don't remember for sure, someone correctly said, oh yeah, transcribing, that's when you write something down. And yes, that is what transcription is. To remind you, that's what the pen is. Transcription is writing something down. And in a sense, it's the same thing with in biology too. We're writing down a code. We're going from DNA to RNA. We're not translating it. It's still the same language, but we're going from DNA to RNA. All right, as you already know, this again is a review too. RNA nucleotides follow the same base pairing rules. So C goes with G, G goes with C. The only difference is instead of there being an A and a T, there is no T with RNA, right? So instead there's a U. You already know that. And again, I think this will be the second time I say this, so hopefully nobody misses this question on the exam. If you get a question where I list a bunch of letters, A, T, T, C, I'll probably list more than that. But if I give you that and I say, transcribe this strand of DNA. Now, if I say transcribe, then your brain needs to say, okay, if I'm transcribing, that means I'm going from DNA to RNA. Therefore, when I'm looking at these multiple choice options, anything that has a T in it is not the correct answer because we are making a strand of RNA, and RNA does not have a T. Any questions about that? Again, that's just a review. Here's the new information. So far, I think this is only the new information I've given you this lecture. So remember, the stuff we talked about way earlier when we talked about DNA replication, the enzyme that does that is DNA polymerase. But now we're talking about transcription. Now we're not talking about making another strand of DNA. We're talking about making a strand of RNA. And the nice, easy thing about this is it's called RNA polymerase. So it's in the name, right? And you do need to know the difference between those two for the exam. But again, it's in the name. DNA polymerase makes DNA. RNA polymerase makes RNA. So hopefully you won't have any problems remembering that. Any questions so far? All right. Here's a picture of how it happens. And again, this should look a little bit familiar because it looks a little bit like the DNA duplication, DNA replication, whatever you call it, because what we mentioned in that situation was that the DNA strands open up so that the code on the inside could be read. And that's exactly what's happening here. The DNA opens up. Here's the code, right? Code's right there. The code's right here. This machine, the cellular machinery called the RNA polymerase, it reads it. It brings in this A. This A is going to hydrogen bond to that T and then covalently bond to that A. This U is going to covalently bond to the A and then, hydrogen bond to the A and then covalently bond to the other A. And it's just going to keep bringing them in one at a time, right? It's going to read the code and bring in the complementary base. So if you understand DNA duplication, this is the same thing, except the rules are a little bit different because, again, for RNA, there is no T, right? There is no T in RNA. There's only a U, and that's it. Any questions about that? All right, moving forward. Oops, you can ignore that. The next word for attendance is going to be 3. I hate those extra lines. Yeah, 3 is going to be the next word for attendance. Anyway, so again, the big picture is you need to know the difference between transcription and translation, which again, transcription is going from DNA to RNA. Translation is going from RNA to DNA, RNA to protein. Once you have that memorized, start adding some details into your brain. And your next set of details is this right here. Transcription itself is broken down into 3 stages, and you need to know them. Initiation of transcription, RNA elongation, and termination of transcription. The good news, in my opinion, is that all these make sense. You know, there's so many things in this class where the names don't make sense. Like, why would you name it that? That makes no sense. This makes sense, right? Initiation, right? That's when you start something. So initiation of transcription, that makes sense. And really, honestly, I put these in bold because for shorthand, you could just remember this. You could just remember initiation, elongation, termination. The other stuff, yes, that's technically the full name, but that will get you. So on the multiple choice exam, I'm not going to say, you know, which of these 3 stages is in the correct order. And then I give you something like, I give you the word initiation, but then that part's different. Or I give you the word elongation, but then that part's different. Or I give you the word termination, but that part's different, right? If you can remember initiation, elongation, termination, you should be good. And again, this should make sense because initiation means to start something. What's the whole purpose of transcription? It's to make a molecule of RNA. So it makes sense that the middle part is elongation, right? You're making it longer. And of course, what do you call it when you end something? Termination. So these names hopefully make sense. We're going to talk about each of these three separately, but for now, are there any questions about this slide? All right. Yeah. So again, my suggestion for your studying is add details, right? So once you have this fact memorized, once you have these three stages memorized, the proper name and in order, then start memorizing some of the stuff I'm about to tell you. Let's talk about the details of initiation and the details of elongation and the details of termination. All right. You need to know where it starts. Just like when we talked about DNA duplication or replication, I said, I told you where on the chromosome it started. Those were the origins of replication. In this situation, transcription is started at a place called the promoter, which in my opinion, that one's not an easy one, right? Because they could have called it the start codon or something like that, but they've got to name it something weird to make it harder to remember. But yeah, it's called a promoter. The promoter is located at the beginning of a gene. So here's a picture of a gene. This purple spot, this is not from your book, the RNA coding sequence. So again, the promoter is right there at the beginning. If you're studying this picture, you don't necessarily need to, because again, it's not from your textbook. So you might see things like 5' and 3' and upstream and downstream, and those are things you don't need to concern yourself with. All you need to know is, where does initiation start? It starts at the promoter. What happens is the RNA polymerase, again, the machine that's doing this, the cellular machinery that's making the RNA strand, it reads this code. And there's something about that code in the promoter where the RNA polymerase says, oh yeah, that's where I need to attach, right? And it recognizes it. Because remember, this is all chemistry, basically. All those A's and T's and C's and the G's, there's something that's attracting the RNA polymerase to it. I will say this, for your own knowledge, not for the exam, because in this course we're trying to keep it simple, but in all actuality, RNA polymerase is not one protein. It's actually a bunch of proteins coming together. And there's a bunch of different versions for it, because that's how your genes work. Because sometimes certain genes need turned on and certain genes need turned off. And one of the ways that we ensure that the proper genes are turned on is by assembling the proper RNA polymerase. So it can actually, how do I put it? It can recognize the proper gene that needs to be turned on. But again, that's just for your knowledge. For the sake of the exam in this course, all you need to know is there's an enzyme called RNA polymerase. That's the thing that does transcription. It attaches at the promoter, which is where transcription starts during initiation of transcription. Are there any questions? All right. Does anybody remember the second one that I know I just told you, so I don't feel bad if you don't remember, but does anybody remember the next step? We have initiation. What comes next?
Elongation.
Good. Was that Cordone?
Yeah.
All right. Can you send me, when you send me your attendance email, please remind me that you did that. I'll give you a little extra credit. Next, elongation, like she said. And it makes sense, right? Because the whole purpose of translation, the whole purpose of transcription is to make a molecule of RNA, right? That's the message. We're trying to make a message, right? So we need to make it longer. That is elongation. It's really simple to understand what happens here. I kind of basically just said it. Obviously, the RNA is going to grow longer. Once the whole process is done, then the RNA peels away from the DNA, right? All the players separate, so to speak, right? Because we're talking about three different parts. We're talking about the DNA, we're talking about the RNA polymerase that reads the DNA to make the RNA, and obviously we're talking about the RNA itself. So once everything is done, everything goes apart, all those three parts, and then the DNA comes back together. Pretty simple, right? Any questions about that? All right, there's not much to say about it. The biggest thing that you need to remember is that it's called elongation, really. Next, termination. Obviously, this is the end. It's in the name. What happens in termination? That's the end. This is the third phase of transcription. So just like you need to know where transcription starts, which is the promoter, you also need to know where transcription ends, which is the terminator. Terminator right there. Not that. I just threw that in there because even though I'm a biologist, every time I hear the word Terminator, I think of the movie Terminator. So that's why that's there. Anyway, at this point, the polymerase detaches from the RNA and DNA. And remember, Oh, excuse me, yeah. Let me back up. Not that it matters because I'm not going to test you at this level, but I don't want you thinking wrong, and I realize I made a mistake. If we're talking about the second phase, if we're talking about elongation, as long as elongation is still going on, I accidentally said that, all three parts were going to go their own way. And that's obviously not true yet because I wasn't thinking. If elongation is still happening, then there's no reason for all the parts to go away, right? That's not until third part, right? That's not until termination. Once termination happens, that's when all the parts go away. So excuse me for my mistake. But of course, like we already said, in the second part, the DNA strands rejoin as soon as the code's being done read. As soon as the code is done being read. Excuse me. I cannot talk today. Anyway, for the exam, the big thing you need to know here is that the third phase of transcription, the end of it is called termination. You need to know that it ends at the terminator. And really, that's it. I mean, yeah, this stuff happens too, but I'm probably not even going to ask you about that. Matter of fact, if you're taking notes, I'll put a big X through that just to help remind you that as far as studying is concerned, you do not need to memorize that fact. Any question about transcription? Pretty simple, right? All right, here's a picture of everything we just talked about. We're looking at a strand of DNA up here. At the top, there's the RNA polymerase. It's just doing its thing. But then it reads the code on the promoter and says, hey, that's where I need to be. So the RNA polymerase attaches right there at the promoter. Then that's initiation, right? Then it starts moving that way. And as it's moving that way, this RNA is getting bigger. And that is what we call elongation. And it just keeps moving that way. It keeps going and going and going until finally, as it keeps going, you can see this RNA strand is growing longer and longer and longer, hence the name elongation. And then finally, it gets to this point, which is the terminator. Once it hits the terminator, it leaves the DNA, the RNA goes its own way, and the whole process is done, at least as far as we're concerned so far. It gets a little bit more complicated, but right now, that's it. So are there any questions about transcription so far? All right, again, the biggest stuff that you need to remember. Transcription is broken down into 3 stages: initiation, elongation, termination. It starts at the promoter. It ends at the terminator. It's producing RNA. The machine that does it, if you want to call it a machine, I keep saying a machine. It's not a machine, right? It's an enzyme. The enzyme that does it is RNA polymerase. There we go. Pretty simple. No questions? All right. So I haven't really said this a lot. So again, if no one knows this one, don't feel bad. But let's see if anybody remembers, where is this happening? Where in this, obviously it's happening in your cells, but where in the cell is it happening? Where is it? Keep guessing. I think you might be on the right track. Is this happening in your, let's see, like chloroplast, mitochondria, your nucleus, your lysosomes, yes, nucleus. And when you send me an email for your attendance, please remind me that you guessed it correct. Yes, this is all happening in the nucleus, right? Because think about it. As far as we're concerned, there are exceptions, but as far as we're concerned, all of your DNA is in your nucleus, right? So yes, this is all happening in your nucleus. Oh yeah, let me say this, and I failed to say this on Monday. It's a good time to bring it up. Of course, it would have been a better time on Monday. But I said in chapters 8 and 9, you can think of your chromosomes like books, right? Instructions on how to build proteins. So imagine this room right here. Of course, you guys online can't see. But imagine this room that we're in, instead of being a classroom, is sort of like a library. And what we have is books on how to build stuff for the university, right? So anytime the people who build stuff and fix stuff in the university need to do that, they come in here. And let's say we have one master copy. Like maybe there's a book in here that says, among other things, it says, this is how you build a bench. So when they need to build a bench out on the quad, they need the instructions. We have the only set of instructions. Like also imagine there's no such thing as the internet or any of that, right? So what do? They need that set of instructions. They need to come in here. Now, can you imagine too, we're just talking about a bunch of people about to build a bench. If they came in and let's say I'm the person in charge and I say, hey, we need to build a bench. I need instructions on how to build a bench. And let's say maybe building a bench is like the instructions are 5 pages long. But I've got this book. Sorry for you guys online. I've got this book that's like this big, thousands of pages, right? And then somewhere in there are the 5 pages that they need. So if they come in and say they need instructions on how to build a bench, am I going to give them the whole book? What should I do? What would you do if someone came in and you've got your one big book, your master copy, and they need to know how to build a bench, and somewhere in in that book are 5 pages that they need? What would you do? What should you do?
Are we talking like?
Yeah, realistic. Well, imagine too, again, there is no copy machines. Let's just say that. Yeah, let's just say there's no copy machines anywhere else, and there's no cell phones. Let's say this is like 1950, right? Yeah, they're trying to build a bench on the quad because they just build things. Just like the university is the cell, right? And this is the nucleus. And they, in the cell, you need to build stuff. And in this case, they need to build a bench. And we've got this big old book, thousands of pages, has instructions on how to build a bunch of stuff. They only need 5 pages because they need to build a bench. And this is our master copy. It's the only copy we have. What should we do when they come in and say, I need instructions on how to build a book? What should we do? Should we give them, here, let's just make it simple, multiple choice questions. Should I give them the book?
I mean, yeah, because we don't have.
I think you were on, I like what you were saying, yes, I guess, because like you said, I think you're about to say because we don't have a copier, right?
Yeah.
True. But I think you were going to say no, right? And why is that? Yeah. Like she said, it's your master copy. That's one of the reasons. It's the master copy. I'm not going to trust a bunch of construction workers. No offense. I'm just kidding about that. But anyway, I'm not going to trust somebody with a master copy. Plus, and on top of that, they don't need some big, bulky, thousands of page book when they only need 5 pages. So you're right. There's no copy machine. But if we could just handwrite it, right? So that's sort of like with the situation with transcription, right? We have the master copy, which is what our DNA is. But outside of the nucleus, it's a dangerous place for DNA, and we'll talk about why later. So when your cell needs to build something, a protein, a particular protein, it's not going to like send the whole DNA outside of the nucleus so you can just read it from there, right? First of all, you don't need the whole DNA. Second of all, you don't want to risk losing your only copy of the DNA, so you write it down. And that's transcription. That's exactly what would happen if they came in and said, I need instructions on how to build a bench. Like, all right, well, let me write it down for you. I'm going to copy it down, and I'm going to give it to you, right? That way, you still have your main copy. They don't have to log around the big book. You don't run the risk of damaging it or losing it. Does that make sense? That analogy makes sense? All right, so that's transcription, and that's the reason for it, too. I should have said that on Monday. I apologize. All right. That being said, now we can throw that analogy completely out of the window for a second and talk about the processing of eukaryotic RNA. So even though we're done with transcription, we're not quite ready to go into translation yet, and you'll see why here in a second. The next word for attendance will be, I'm going to circle it and not say it this time, as you know I do at least once in a lecture. That is the next word for attendance that is circled. That being said, let's talk about this. First of all, I want to remind you, because we skipped chapter 4, so now's a good time to remind you, as I always do every time this comes up, we human beings and plants and fungi and other animals, we human beings, other animals, plants, fungi, we're complicated structures, right? We are what we're called eukaryotes. So life is broken down into 3, but we don't get into that in this class. But let's just say life is broken down into 2 major types, eukaryotes and prokaryotes. Prokaryotes are things like bacteria. They're the really basic ones. They don't have organelles like nuclei and chloroplasts and mitochondria, things like that. So with that reminder in mind, in prokaryotes, which are things like bacteria that don't have nuclei, the RNA that is transcribed from the gene immediately functions as the messenger RNA. The most important thing in that sentence, that bullet point, is this right here, messenger RNA. You need to know what messenger RNA is. So basically, every time I've used the word RNA so far in this lecture, in this chapter, I've actually been meaning messenger RNA. But this is the first time I've specifically called it messenger RNA. And that seems easy to remember because right now that's the first one I've given you. But the thing is, there's actually going to be other types of RNA that you're going to need to know about. So yes, messenger RNA, you need to know that one. It's in the name, right? It's in the message. So again, if the construction workers are coming in because they need instructions on how to build a bench, that's what I'm writing down, right? I'm writing down the message, all right? Here's how you build it, right? Writing down the message or the messenger, right? And that's what they're going to use. So yes, you need to know what messenger RNA is. And I'm going to put it next to the rest of this. Your book is just given a little bit of, I don't even know why they included that. It's true. In prokaryotes, because there is no nuclei, because the DNA is all right there in the cell, then as soon as you make the RNA, it's ready to go. But that is not the case with eukaryotes. We're a little bit more complicated. First of all, we have a nuclei, right? So things need to be ready before they can leave the nucleus and go into the cell. Again, like you already know, if we're talking about eukaryotes, which is us, which is what you're going to be tested on, which is, again, why I put an X through prokaryotes. Transcription happens in the nucleus, as Cordone already properly said for some extra credit. But then, and remember, transcription is just making this strand of mRNA. Then, after this happens, the RNA has to be modified before it moves into the cytoplasm. And another word, another statement, another way of saying into the cytoplasm is out of the nucleus. It's all the same thing, right? Cytoplasm is basically in the cell. In this case, it means outside of the nucleus. Of course, if we were talking about respiration, in the cytoplasm means outside of the mitochondria, right? It's all a relative term. But anyway, in this case, it means before the mRNA can leave the nucleus, it has to be modified. And really quickly, I'll tell you why. There's a lot of reasons why. And you're really going to learn about this here in the next few minutes or the next section of this chapter when we talk about viruses. But your cell does not like DNA and RNA that doesn't belong in your cell. If it sees it, it tries to attack it because usually that's a virus and that does not need to be there. So what happens is if your RNA were to just come floating out of your nucleus without protection, your cells would probably destroy it. And on top of that, there's also more cellular machinery that actually does the translation, which we haven't talked about yet. And part of what we do in this process is getting it ready for that. So it's all about protection, and it's all about getting it ready for the next step. That being said, let's talk about it. Let's talk about how it changes, what we do to it. And you need to know this. The first thing that happens for the processing of RNA before it leaves the nucleus is something called cap and tail. It's pretty simple. Well, the way we teach it is pretty simple. You get a cap, you get a tail. Meaning, yes, you have that RNA that's exactly as was written from the code in the DNA. But then at the end, we also add some extra bases at the end. The cap is one thing. We won't even talk about that. The reason this is called a poly-A tail is because it's literally just a bunch of A's. A, right? We're just adding more nucleotides to both ends. Again, like I was saying earlier, why do we do this? Well, it protects it from attack by enzymes. So we're talking about protection because, again, your cells don't like foreign DNA and foreign RNA in the cell. So you've got to protect it. This kind of helps it recognize it as its own. Like, hey, don't kill me. I'm one of you. And then again, like I said, there's cellular machinery that actually does the translating. And what we're doing here by adding this poly-A tail and the cap is helping that cellular machinery recognize that and say, okay, this is the thing that I need to translate into proteins. And you don't have to write it down yet because we're going to talk about it a little bit later, but that cellular machinery is called ribosomes. So again, what you need to know for the exam is RNA, the mRNA does not leave the nucleus right away. It has to be modified. One of the ways it's modified is something called cap and tail. And if I ask why, I'm going to put a question mark because I'm not sure I'm going to get that deep on the questions. But if I ask why we do the cap and tail, what's the purpose? You could just remember protection and ribosome recognition. But I probably won't get that deep with the questions. So any questions about the cap and tail? This picture is not from your book. You don't need to memorize the fact that the cap is on the 5' end. I haven't even told you what 5' means. You don't need to memorize the fact that the tail, the poly-A tail is on the 3' end. You don't even need to memorize the fact that it's a poly-A tail. It's just a tail. For you, cap and a tail. That just means for you on both ends, we just added extra nucleotides. You don't even need to know which end is which. Just know it's for protection and for ribosome recognition. Any questions? Okay. So again, before mRNA can leave the nucleus, it has to be modified. The first one we just talked about was cap and tail. The second one we're talking about is RNA splicing. It's another type of processing. What this does is removes non-coding nucleotides from those that do code for amino acids. And this is new information to you. I haven't told you this before. But when you're looking at a strand of DNA, it's not, or a gene, if you're looking at a gene, not all the codes are actually useful. This is stuff we used to call junk DNA. We don't call it junk DNA anymore because now we know that it actually does serve a purpose, depending on what it is. But it doesn't serve the purpose that we're talking about here, which is producing proteins. So that stuff is called, excuse me, those things are called introns, and you need to know what an intron is. So that is those are the parts of the DNA, or in this case, the RNA, because remember, we copied the DNA into the RNA. Those are the parts, the introns, that do not code for amino acids. Therefore, the other parts are called exons. Those are the parts that do code. I hate the nomenclature on this. When I was I was never young as a student, but when I was early into my academic career, I got those two confused. Here's how you need to remember it. The exons, that's the part that's being expressed. That's where the X in the exon comes from. That's the part that's being expressed. That's the part that has the code for the amino acid that's getting expressed. The other stuff that doesn't do anything, that we're going to call junk DNA because it'll help you remember, but it's not actually junk DNA. That's introns, and where it gets its name is it is in between the useful stuff. Introns are the in-between stuff. Exons are expressed. The introns are the in-between stuff. So again, it's the introns that we get rid of. It's the exons we keep. And that's the part that confused me as a student, because to me, it sounds like you keep in the introns and you get rid of the exons, right? In the name, it sounds like that. So just try to remember, exons are expressed and we keep them. Introns are the in-between junk that we get rid of. And we're going to talk a little bit more about it, but are there any questions so far about RNA splicing? All right, so this picture here, it shows you. Here's pre-mRNA. So again, this just came off the DNA, right? The RNA polymerase just made this. It's got its cap and tail, which it doesn't show here. It's getting ready to leave the nucleus. It needs to go through RNA splicing. So it gets rid of all, the introns. In this case, there's two of them, and they're colored blue, right? So the introns are gone because that's the in-between part. That's the useless stuff. So just kind of, making it more streamlined, so to speak. So any questions about this? Oh yeah, I have another analogy that might help you remember this. So imagine we're going back to the book, right? This is the building that has all the books where you can construct stuff. But let's say also, what's another thing we have on this campus? We have a cafeteria, right? So one of the things that we make on this campus is food. That is making something. So let's say amongst our books of how to build stuff, we also have recipe books on how to make stuff. Have any of you guys ever looked up a recipe recently online for anything? Anybody online or in person? Yes. How about you online? Has anybody ever looked up a recipe recently on how to cook something?
Yeah. Yes.
Do you get the same garbage I do? We get a whole blog, like all I want to know is how to bake chicken, and like my grandmother.
Yes.
She had great memories of summers in Vermont, and we used to bake chicken. And I don't care, just show me the recipe. That would be the introns, right? That would be like if I had those recipes and the people came in here and like, look, I just need a recipe on how to bake chicken. I'm like, all right, well, let me sit you down. First, I got to write to you about my grandmother's upbringing in Vermont, right? You don't want that. You get rid of all that junk, get to the point. That's what this is, right? Get rid of the junk, get rid of the introns. Just give me the stuff that's expressed. Just give me the exons. Any questions about this slide? Again, we're going to talk a little bit more about this, but are there any questions about this slide? Hooray. All right. As I've already mentioned this, but obviously these exons and introns didn't come from nowhere, right? They were transcribed from the DNA. They didn't come out of nowhere. The RNA polymerase read the DNA and copied it verbatim. right? So that's why exons, introns, it was all there, junk and everything. But again, like I've already said, before the RNA leaves the nucleus, those introns are removed, right? We want to get rid of that junk. And then, obviously, if you get rid of this stuff, you have to join the exons. Otherwise, you would just have a bunch of RNA pieces. So obviously, the exons have to be joined. I'll go ahead and put an X through this. I'm not going to ask you that detail. But it is obvious, right? Literally, if I had scissors right now and I just cut this and then cut this, I would have 1, 2, 3 strands of DNA or of RNA. We don't want that. We want one strand. So obviously, you would have to splice them together. Now, if any of you have ever done electronics, this is actually what I did in the Marines. I was an aviation electrician. So this is actually stuff we used to do. If there was a problem with a section of the wire, we would cut off the bad section and we would splice the good sections together. That's where it gets the name splicing. It is something that is done. That is a word that has meaning outside of biology. Anyway, we still have a little bit more to talk about RNA splicing, but are there any questions so far? Okay. So here's where it gets a little bit interesting. I'm going to put an X through this, though, because even though it's interesting, you could go the rest of the semester without understanding this and be okay. Therefore, I'm not going to test you on it. RNA splicing plays a significant role in humans, and it allows our 21,000 genes, and I don't remember if it's exactly 21,000, or it's definitely not exact. I don't remember if it's 21,000 anymore. This was a few years ago. You can look that up for independent work if you want. Really quick, easy sentence. How many genes are there in the human genome? Anyway, the point here, though, is we definitely have many more proteins being built than we have genes. And when you think about that, that should make no sense to you, because genes are instructions on how to build proteins. So let's assume that number is exactly correct. Let's assume we have 21,000 genes. And then I'm going to make up another word, or another number, just to keep things simple. Let's say there are 50,000 polypeptides, aka proteins. How do you explain that? If a protein can only be built by using the instructions written on a gene, then how is it that we can only have 21,000 genes yet have 50,000 polypeptides? And the numbers aren't important. The fact is we have fewer genes than we do polypeptides. Those numbers are just, I threw them out there. So can anyone guess, especially in the context of what we're talking about, how can that happen? How can we have more proteins than we do genes that provide instructions on how to build proteins? This is a tough one, but don't feel bad if no one gets it. But no guesses? All right. I will show you. It's called, well, no, not yet. That picture should have been earlier. All right, let's back up a second. Everything we talked about with RNA splicing so far, there's a picture of it. There's your DNA up top that includes all the junk too, right? It's got the exons. It also has the junk introns. It has it all. The RNA polymerase doesn't know any better. It copies it verbatim. So that first version of the mRNA, it includes all the junk. But again, we don't need it. So the junk is removed. Those RNA pieces are spliced back together. So now we have one piece. Now it's ready to go. It leaves the nucleus and goes into the cytoplasm, and we'll talk about that later. Now, going back to this idea of 21,000 genes, yet 50,000 polypeptides, how does that happen? And this is how it happens. It's called alternative splicing. So here's another picture, not from your book. Here's another gene. It's got 1, 2, 3, 4, 5 exons. And the real gene has many, many more than that, but this is just keeping it simple. Therefore, this part's not labeled, but that means this is an intron, this is an intron, this is an intron, this is an intron. Now, what I first talked to you about was this right here. When I said RNA splicing, I said we got rid of all the introns, right? We got rid of 1, 2, 3, 4 introns, and that would bring together this exon, right? That's what this is. However, there is alternative splicing. So in this version, we do the same thing. We always get rid of all the introns. But in this version, not only did we get rid of all the introns, we also got rid of exon number 3. And in this version, not only did we get rid of all the exons, but we got rid of intron number 4. Excuse me, exon number 4. So because of this, you have different strands of mRNA. all from the same original code of DNA. So from the same original gene in this simplified version, we now have 3 different proteins being built from the same gene, all because we left off some of the exons. So, and this is just a picture of what it looked like. You can see protein A, protein B, protein C. Those pictures probably make no sense to you, and that's okay. They don't need to. But you can tell this thing looks different than this thing, which looks different from that thing, right? And remember from chapter 3, a protein's function is all about its shape. And even without being an expert, you can look at those things and tell they have different shapes. Therefore, they have different functions. Therefore, they're different proteins. So that answers the question, how do we have only 21,000 genes, but 50,000 proteins? This is how. Because one gene, when it's alternatively spliced, can make multiple proteins. And again, this is a very simplified version. There's many more exons than this in your typical gene. And sometimes, too, it'll be like, I won't get into it. Let's just say this is the very simplified version, and it gets much more complicated than this, and you can have many more proteins from the one gene. But any questions about alternative splicing? All right. Good. This will be perfect. Let's talk about translation next. The translation is a lot more complicated than transcription. So much so that your book breaks it down into 2 parts. First, before we even talk about the process of translation, your book just tells you the parts. Like, all right, here's the players, as they call it. All right, here's the things that we use. And then once you're familiar with what's going to be used in the process, then we talk about the process. So hopefully today we can talk about the parts, and then hopefully Friday we can talk about the process and then viruses and be done. Oh yeah, that's a good time to talk about this then. That would mean, if that happens, if we finish chapter 10 on Friday, that means the exam will be next week. So you guys start thinking about that. Send me an email if you have a particular time you would like, or a particular date you would like to take the exam. If Monday or Wednesday or Friday works better for one of you, let me know. And I guess the squeaky wheel gets the grease, so to speak, right? So, no, this is just exam 3. Good question. Final exam will be about a month from now. All right, translation. The next word for attendance will be machine. That reminds me of a stand-up comedian. I won't get into it. The machine. Anyway, again, we're talking about translation now. We're done talking about transcription. We're now talking about translation. We're talking about going from RNA to DNA. Specifically, that RNA we just got finished talking about, the one that got the cap and the tail, the ones that we got rid of all the introns. We're talking about translating that into protein. And I said DNA again, I think. I apologize. I don't know why I keep doing that today. We're talking about translation. We are talking about going from RNA to protein. Man, I've got to quit screwing that up. Anyway, again, like I said earlier, before we talk about the process, we're going to talk about the parts, the players. So here's the first important part, because we've been talking about it already. It's the mRNA itself, right? The mRNA, that's the code. That's the message. That's what the M stands for, right? Messenger. So this is the part. This is the stuff that's going to be translated. This is the code. Or going back to my analogy of we had that big book and somebody needs to build a bench, right? This is the part where we wrote down how to build a bench. We left the whole book. They don't need it. Now the construction workers have that set of instructions, handwritten instructions on how to build a bench. That's the mRNA. That's the first part. The second part is the machine that's actually going to do the translating, the thing that actually reads the code and builds the protein. And we're going to talk a little bit more about that later. But just for now, know that there is a machine. And just like in DNA duplication, it's DNA polymerase. Just like in transcription, it is RNA polymerase. And then translation, there's another word for a different machine, and you'll find out what it is soon. Obviously, too, this is going to require energy and enzymes and energy. We'll talk about the enzymes soon. And I'm going to put it next to this, not because it's not important, just because I'm not going to test you on it. Obviously, it requires energy. Anything we do, actually, same thing with enzymes. Anything we do in your cells is going to require enzymes, it's going to require energy. And obviously, it's going to be through ATP. I put a little star next to that because you can also get energy elsewhere, but we don't talk about that in this course. Translation also requires 2 other components. But this is stupid. I'm sorry. I'm looking at this slide now. I need to fix this. Let me put a big arrow here. The machine that translates the mRNA, that's the ribosomes. I don't know why I laid it out like this. Here's some new information. There's also a different kind of RNA called tRNA, and I'm going to talk about that in a second. So for now, if you're taking notes, you don't need to write down tRNA because I'm going to tell you what it is. So yeah, we're about to talk about all the parts individually, but this is all of them listed at once. mRNA, ribosomes, tRNA. Well, there's one more component, but we'll get there when we get there. But those are the big ones. Any questions about this slide? All right. Yep, like I already said, it requires an interpreter. I keep using different words. Interpreter, in this case. Actually, I'm just going to put a line through that. We'll talk about this, but I want to make sure I'm talking about it in the correct context. So you just forget about that. You'll get it in a different context. What we need to do in this process, to throw it back to what we talked about on Monday, we need to convert the 3-letter nucleic acid words, which are called codons. Remember, you need to know that. So remember, nucleic acids, if we're talking about RNA, there's U's, whoops, A's, C's, and G's, right? Those are the 4, that's the alphabet of that language, so to speak. And in that language, there's words. And those words are always 3 letters long, right? So it's some version of U, A, C, and G, some combination of them, 3 letters long. So those 3-letter words have to be translated into amino acid words. And this part right here, I'm going to put a check mark to it to remind you this is what we talked about on Monday. You have the code that says, bring me this amino acid. All right, thank you. Next, bring me this amino acid, and we'll attach it to that one. Next, bring me this other amino acid, and we'll attach it to that one, right? That's all the codes are. So it tells you which amino acid to bring. So what does it? What brings the amino acid? We already know what the code is. That's the messenger RNA. But I keep saying, we need to bring me the amino acid. So what is it that brings me the amino acid? It is the transfer RNA. And you need to know that. And like I said, there's going to be different RNAs that you need to know about. So this is the second one you've learned about. Transfer RNA matches the amino acid to their codons and forms a polypeptide. I'm going to put a box around here because if you're studying, if you can just memorize this part, you're good. The rest of it would be good to know. But if you can just memorize the fact, it's the transfer RNA that's bringing the amino acid. So if I am a ribosome, if I'm the thing doing the translating, and I don't know, let's say he was a tRNA. What I would do is I would read a code. I'd be like, all right, bring me a tryptophan. And he'd be like, okay. And he'd go get a tryptophan, and he'd bring it to me. And then I'd say, okay, bring me a serine. And then let's say she's a tRNA, and her job is to bring serine. So you'd be like, okay, and she'd go find it, and she'd bring it to me. That's how it works, right? Basically, the ribosome reads it, says, all right, I need this amino acid. And then the tRNA says, hey, that's me. And it brings the amino acid to the machine. This will make a lot more sense when I show you the picture. Therefore, the tRNA carries out 2 functions. Obviously, it picks up the appropriate amino acid. So just so you know, it's not like there's one tRNA that brings all the amino acids. It's actually more specific. There's one tRNA for each amino acid. That's why I was given the example. I said, you know, if he was a tRNA, I would say, bring me a tryptophan. And he'd be like, okay, that's me, because he's the tryptophan tRNA. And I said, bring me a serine, and that would be her. She's like, oh, that's me. That's what I carry. So anyway, yes, it picks up the appropriate amino acid, and then it recognizes the appropriate codons in the RNA. I'm also going to put an X through this too, because I just want to help you guys study. Yes, that's important information, but I'm not going to test you on it. The big thing here that you need to know is that the transfer RNA is the thing that's transferring the amino acids to the ribosomes. Oh, shoot, it's 8:51. That's it. All right, if you were not online for lab on Monday, and I haven't sent you an email saying you don't need to come to lab, then you need to be online for lab today. Because as some of you know, if you're absent, you just get quarter credit for doing your lab. And that hurts. That hurts your grade. So don't be absent, please. And sorry I kept you late. You guys send me emails for being here, please. And that's it. Yes. I'll take it to my office. I had to get out of here.